Good afternoon, everybody, and thank you for joining us in the CUNY Grand Rounds seminar today. My name is Elias Kavouras, and I'm the Chair of Environmental, Occupational, and Geospatial Health Sciences. Couple of housekeeping items. This presentation is recorded and may be available sometime later. All of you are muted and your video is, you're not allowed to switch on your video. If you have any questions, please post them on chat, and then we'll follow with a Q&A session at the end of today's seminar. So without further ado, it's with great pleasure today to introduce you our speaker, Dr. Matthew Campen. Dr. Campen is the Regents Professor in the Department of Pharmaceutical Sciences in the College of Pharmacy at University of New Mexico in Albuquerque. He received his PhD from the University of North Carolina at Chapel Hill School of Public Health and completed a postdoctoral fellowship at the Johns Hopkins University School of Medicine. He has continuously funded by NIH since 2007 and has funding for the foreseeable future, and other federal foundation and commercial sponsors. He co-authored over 130 peer-reviewed publications in air pollution health effects across a wide range of experimental designs and approaches to better understand exposures, the biological mechanism of action, and health outcomes. Dr. Campen directs the new National Institute of General Medicine Sciences, funded New Mexico Center for Metals in Biology and Medicine, and he's also the co-director of the UNM Clinical and Translational Science Center. Today, he will talk about the neurovascular effects of wildfire smoke. You remember these hazy orange smoky days last summer that we experienced so intense? It's not only something in the West, it's coming to East, reminding us that climate is changing, and it changes everywhere. So without further ado, Matt, I would like to welcome to our Grand Rounds.
Thank you very much, Elias. I really appreciate this opportunity to share our work. We were joking before about how much more interested the media of the United States has become since the wildfire smoke descended on the East Coast. And obviously Canada suffered a massive fire season this year, and literally 100 million people across the East Coast had really, really bad air quality for days to weeks. And so it stands to reason that there would be a lot more attention to this. I'm going to share what we've been doing over the past 5 years or so on this topic. Most of my research is in preclinical models, meaning mice, and some cell culture work. It's relatively basic science, so I apologize in advance if it's a little on the more molecular side, but I'll try to bring it back up. But there have been some very nice epidemiological studies in the past couple of years that we use really as anchors to help understand the results we're finding and put it in the context of a real population level effect. So yeah, we're going to talk about the blood vessels of the brain and how they're really the interface of toxicity to the brain that occurs indirectly from the inhalation of wildfire smoke. So I love these maps. This is actually an app I have called My Radar, and I don't get any money from My Radar for using this, so no conflicts here. But there's this, and there's other ways of visualizing the dramatic distance that wildfire smoke can cover. So these were the fires about a month ago up in the Pacific Northwest, and you can see that they blanket more than half of the continent, affecting, I mean, there's almost 400 million people in this country, and most of them are on the East Coast. So that's a huge number of people being impacted by this deteriorated air quality. What I like about this image also is if you want to know how weird the world has gotten in terms of climate, the reason that the Mountain West region isn't blanketed by this fire smoke is that there's a remnants of Hurricane Hillary sitting in the middle of Nevada, which is about the oddest thing I've seen in 10 years or so. So that's where we are. The broader trend is that, yes, wildfires are increasing in size over the past 40 years. Not number, the number of fire events are about the same, but the extent to which they destroy acreage has increased almost threefold since the early 1980s. Most of that acreage has been on the Pacific Northwest and Mountain West states. And unfortunately, the flow of atmosphere is from west to east. And so the smoke that comes from those fires travels across the population of our country. This is probably a global issue. It's not just us. I was not able to find really great data, and this is a really poor graphic that I pulled up and expanded just to make this simple. But we think that this is overall a global problem where you have increased severity of aridity, meaning the dryness of certain regions has been enhanced because of climate change. And as a result, you have areas that were previously rich for growth of plants now becoming drier, and you have that risk of dramatic fire and large acreage damage. This is a map of New Mexico. This was last year. We set a record. There were large fires down in the Gila Mountains close to Arizona. Not many people live down there, but up in the north between the cities of Santa Fe and Las Vegas. We have our own Las Vegas, by the way. It's not quite as large or showy as the one you've heard of. But we had about 200,000 acres burned up there in the Cap Creek Hermit's Peak fire. And that looked something like this, where, we had two fires form, and they eventually came together, the Calf Canyon and Hermit's Peak fires, and they were intentional burns that got out of control. So again, it's not the number of burns, it's the severity and the extent to which they burn. But basically, 15,000 people in Las Vegas had to evacuate because the smoke and risk of fire was so bad throughout that period. So what is it in the wildfire smoke that we worry about? It's a combustion mixture, and there's lots of particles and gases. Particles are something we've been studying for over 100 years, honestly. We've known that smog can be deleterious to health and well-being. We've known that heart and lung problems can occur, dating back to London fog incident of 1952, and even before that, there were some smog episodes in Pennsylvania and Belgium. But the smoke that we worry about, the particles in the smoke that we worry about are very, very small. We're talking about stuff that's less than 10 micrometers in diameter, or even smaller. We talk about PM 2.5. These little tiny droplets, and the reason that small matters is that the smaller they are, the deeper in the lungs they can get. The deeper in the lungs they get, the longer they stay in the lung. That's, to me, the biggest issue is they get down deep, they sit in the lungs, and the mucociliary tract of the lungs will pull them out, and you'll cough them up or you'll swallow them. But it can take weeks to get those things out. And so people that are vulnerable with, if they have asthmatic conditions or COPD or something like that, they can suffer effects days to weeks after the exposures. And I like to point out that particulate matter, PM toxicity, is a combination of both a physical insult as well as the chemistry, because you can have dissolved metals, but you can also have certain shapes. I like to point out asbestos has a very jagged, pointy shape that is very difficult for macrophages to destroy. So the size and shape is really crucial there. But additionally, there are gases in the wildfire smoke, and they're all different kinds. I think most of it is going to be carbon dioxide and carbon monoxide in terms of what comes out, but you'll also have a lot of volatile organic, semi-volatile organic species. And I like showing off this study from Young Ho Kim and Ian Gilmore down at the EPA in North Carolina, just because what I like about these graphs is these large gray bars at the top is unknown. So it's very difficult to get a grasp of all the different chemicals, even with some high-end mass spectrometry readouts. but basically there's a combination of different shapes and forms of organic material or elemental carbon that come out in the gaseous material, and that can then bind to and interact with particulates. And so that's acutely, that's right next to the exposure, but these exposures, of course, travel thousands of kilometers. So the question is, what are we really exposed to at a long distance away? This is a picture from the Sholo fire in Arizona from 2011. And you can see how it's all flowing in sort of a direction that's based on the prevailing winds at that time. Sometimes this would travel north of us, and sometimes it would come right on top of Albuquerque, which is, I want to say, very much due east. And we would see ash fall and particulates in the air and smog. And so when this happened back in 2011, I grabbed data out of the Metropolitan Monitoring Group, the Air Quality Board. And what we were able to show is that if you look at particulate matter levels from May to June of that year, you can see that it spikes very plainly during that wildfire period, and it comes and goes. It's very intermittent depending on the meteorology. Also, we saw that carbon monoxide levels came and went with those spikes of particulates. That is interesting because that means that the carbon monoxide is a good marker. The overall levels of carbon monoxide were not that high. They were on the order of 1 ppm parts per million at the peak. The regulatory levels for long-term carbon monoxide exposures is 9 ppm, so this is well below that. But it's such a clear marker. You can see that it goes up on those days. And it travels well. Carbon monoxide is a very stable molecule in the air. It's a very stable molecule in your body. If you breathe in too much carbon monoxide, the only way to get it out is to add a lot of oxygen through a hyperoxic tent and force it out. But it comes out as carbon monoxide. It's not like it gets metabolized. But then some of the other gases like oxides of nitrogen or ozone, these are common pollutants that are monitored by the Air Quality Division. They really didn't show the spikes on those days. And those are because the oxides of nitrogen are formed from wildfire smoke, but they degrade, they react out very rapidly. And then ozone is actually a regional product or a local product that is dependent on local emissions on your roadways and interaction with sunlight. So I just like to show this because it really reveals the trends for long distance travel of these smoke episodes. So in my research group, we're not disinterested in what happens in the lungs, but for the most part, we're happy that there's a lot of people researching how wildfire smoke and other pollutants affect the lungs. And my interest has really been on some of the things that happen beyond the lungs. Among them, incidents with cardiovascular disease. We've really gotten into neurological diseases, neurodegenerative, as well as developmental learning and memory. And that's been a force over the last few years that has driven us forward. We've also been looking at maternal fetal effects. I won't talk about those, but that's definitely another interesting area of research. I'm going to quickly go through what we think is sort of a mechanistic pathway for how you have indirect effects beyond the lung, because your lung is not an active sieve that just lets all this stuff through. It's actually a very effective barrier. You have a mucociliary escalator that takes most of the particles out and sends them through your GI tract. You have lots of immune cells that eat the particles and take them up that way as well. You even have a lot of metabolizing enzymes like P450s in the lungs. So your lungs are a beautiful defense mechanism. So how it is that you end up with responses in the brain, and I'll talk a lot about this vascular inflammation that ensues. We've been tracking that down over the last decade, and this is sort of the snapshot of the story I'm going to tell you. And we work with a lot of different air pollutants, including these multi-wall carbon nanotubes, these purpose-engineered materials that are exposed to occupational populations. Ozone, which is just triatomic oxygen. It's a very common environmental pollutant, but it's also a very nice, simple pollutant that doesn't get past the lungs because it's so reactive. And then we work with other complex mixtures and combustion sources, like this is a diesel particle floating around. It's very interesting, made up of lots of little tiny particles, but ultimately behaves like a slightly larger particle. So our studies have shown that the lungs have a fairly common range of responses to all of these things that involves activation of the innate immune system. Matrix metalloproteinases are among those most crucial enzymes that are activated. And when they are activated, they start chewing up other proteins, proteins especially like extracellular matrix that's all throughout the lungs. And the result of degraded or broken down proteins is little tiny peptides. And so this is work of my colleague Andrew Ottens, who's at Virginia Commonwealth. And what he's shown is that in the serum of individuals or laboratory animals who are exposed to any of these toxins, you end up with a whole host of it. I've called it molecular shrapnel, but they're broken bits of proteins that surge after these exposures. They eventually return to normal after, hours to days to months, depending on the protein or the individual. But you can then follow the amino acid code of each of these peptides and link it back to the origin of the protein. And so we've seen that, this is sort of an ontology classification scheme. This blue box shows extracellular matrix peptides. There's also a group of peptides that would be associated with cardiovascular changes or inflammation. So this is very real. And what happens is these peptides, they don't look like their original protein entirely, but they still retain some characteristics, including binding regions. And so they actually still have some bioactivity. They're circulating, and they can bind with a number of different receptors on endothelial cells to cause changes. And so we had done a study back in 2010 to 2012 where humans were exposed to diesel exhaust. This was with folks at the EPA in Chapel Hill. And we got serum before exposures, after exposures, and 24 hours later from diesel and nitrogen dioxide. And what happened is we took that serum in a dilute fashion and put it on endothelial cells and let the endothelial cells tell us what they thought of the serum. And what they thought was, after the exposure, that serum was inflammatory. And here we're measuring the expression of vascular cell adhesion molecule. We had also looked at chemokine CCL2 as well as ICAM1, and they all behave the same way, where both diesel and nitrogen dioxide cause this acute increase in the inflammatory potential to these vascular endothelial cells. And even 24 hours later with diesel, that was still elevated. But the filtered air control group, which were the same people it was a matched repeated measures study, that didn't cause this effect at all. So there's very clearly something that's inflammatory in the mixture. And when that gets to distal organs like the brain or the placenta, you have activation of those endothelial cells. And here we're showing in the brain that you can have a loss of the blood-brain barrier integrity. And so the green, these are cross sections of blood vessels. And, in the middle is where the blood should be. And we're staining for albumin. And hopefully you can see there's very little green on the outside of the blood vessel in the control. But in the exposed, there's quite a bit of green beyond the barrier there. And also you see that when that barrier breaks down, the brain has to respond with activation of its immune support cells, including the glia, the microglia, as well as the astrocytes. They come in to shore up the defenses around that blood vessel. And when they do that, they can't do what they're normally supposed to do, which is a whole host of things, but certainly involved in learning and memory. They're certainly involved in clearance of waste products from the brain and potentially neurofibrillary tangles, tau related or A-beta related plaques. So that's sort of the big picture of what we're studying. And this I just like to come back to the endothelial cell, how important endothelial cells are throughout your body. So if these molecular shrapnels, these peptides coming from the lung, interact with the endothelial cells in any organ of your body, they can be triggering increased inflammation and leukocyte trafficking. They can alter hemostasis and coagulation. They can change vascular tone. So one of the things we saw early on was a hyperconstriction of blood vessels that, if you're having a coronary event, could be very detrimental and make the outcome far worse. And we've known about this for different particulates for a long time. But now we're at the stage of identifying that wood smoke in particular and wildfire smoke may be specifically damaging. So some of the work I'd done in a previous life, we had been looking at those endothelial cells in vascular preps after exposures to all kinds of different pollutant atmospheres. And what we could see was that if you look at the hardwood smoke, we've stained specifically for activation of metalloproteinases. And you can see that right along that endothelial cell layer, there's this sort of glowing that shows, yes, it is in fact these endothelial cells that are lit up as a result of these exposures. And I'm just going to move on with that story so I can get to the fun of some of the recent work we've been doing. 2 years ago, well, 1 year ago, this paper came out from Anna Rapol's group, and it was a population level countrywide study where they had a brain training app. They had data from this brain training app, and they had multiple data points for 10,000 individuals throughout the country. And you can see that the top map shows the number of gamers from a certain region, and you can see that certainly around the Salt Lake area and Denver and Phoenix, there was a strong representation of people out west, Texas. They had a lot of people all over the country taking this. And at the same time, they were able to look at the geospatial distribution of particles coming from wildfire. And when they overlaid the data and they analyzed for the scores on that brain training app, what they found was that there were very clear impacts both on the attention score as well as the accuracy score. So, analogous to cognition as well as focus and attention, right? And what was interesting was that their effects seemed to be more dramatic a week after the exposure compared to just 1 day later. So this is an enduring effect on the brain that is population level based on these exposures to wildfire smoke. So we decided to look into that. So this is our first study that I'm going to talk about. This is published a couple years ago in Toxicological Sciences. And we had already been working out in Navajo Nation and Laguna Pueblo using this mobile laboratory. It's a 53-foot trailer to capture particulates and expose animals, mice, inside the trailer to exactly what is in the air around us. And the reason we were on these sites was there was concern from the communities that uranium mines in the area might be distributing dusts that contain uranium or other heavy metals. Long story short, for the uranium story, we did not see much. There was very little reason for concern, and we've shared that with the communities. It's been a really wonderful story in that regard. But while we were out there, we noticed a few trends. Here's the trailer again. This is out in Blue Gap Tachi, Arizona, right in the middle of Navajo Nation. When we looked at collectively a bunch of different exposures that had happened, what we found was that there were exposures done in November and December versus exposures done in April and May. And in November and December, in this little low-lying canyon, there were a lot of households that were burning wood smoke for heat and cooking in their house. And even though the overall wood smoke levels were lower during the winter compared to the spring, where there's a lot of wind-blown dust, we have really windy spring times. Despite that, the macrophages in the lung and the neutrophils in the lung were much higher, significantly higher from the lower concentrations. And we could tell you anecdotally that when we would walk around on those days, we could smell the wood smoke. We noticed it was there. There was a little bit of inversion. And so that sort of got us thinking, well, all right, we've got this trailer parked. This was in Laguna Pueblo in the village of Powati. It's very close to Mount Taylor, which is one of the 4 sacred mountains of Navajo legend. And while we were there, towards the end of one of our other exposures, we noticed that there was this huge wildfire outbreak in California. And we thought, let's get mice out. Let's get this started and see if we can do a real wood smoke exposure. And so on the subsequent days, we saw that wood smoke dissipated throughout the big valley there in California and started moving west. And you can see it's lighter, it's dilute, but it did cover across Albuquerque. We also had some wildfires popping up in Colorado and Arizona that may have contributed. And then I like to show this image just because after 20 days of exposure, we stopped our exposures. And if you read the paper that we wrote, you will not really get an understanding as to why 20 days was important. Basically, because on day 21, it snowed. And we decided we had to stop the exposures out of fear for our personnel driving in and out of these conditions. So just so you know, sometimes research goes awry and you just have to roll with the punches. So we did a 20-day exposure, and we were able to very cleanly show that on the days with the high particle levels in the exposure chambers, we did have evidence that wildfire smoke was on top of us in high representation. Mice were exposed daily, 4 hours a day. The rest of the time, they were just in their home cage, relatively filtered air, clean conditions. We also used levoglucosan as a marker of the particles to show that there was a wildfire or wood smoke contribution. But overall, for those 4-hour periods, the concentration average was 104 micrograms of particles per cubic meter. That is above the EPA recommended levels by far. I think the EPA level now is 12 micrograms for a 24-hour period. And keep in mind that we only exposed for 4 hours. So if you divide 4 by 24, you can divide 104 by 6, and you get down to around 20 micrograms. So it's not that much higher than the 24-hour. But this is even more interesting. These are the 24-hour average, really 13-day average exposures for all of Washington state, where each dot represents a single county. And this was a 13-day air pollution episode from wildfires that they saw back in 2020, about the same time we were doing our exposures. And I've just re-represented the data based on population distribution. So you can see that in the entire state of Washington, you have 1.6 million people that are constantly exposed to levels greater than our exposure level, and 5.5 million to between 50 and 100. So obviously you can get indoors to escape that, but there's probably a lot of incident exposures as well as houses that don't filter very well. So the exposures we're working with are very, very much realistic exposure concentrations. So after these exposures, we examined the mice for the degree of inflammation in the brain, and we did this using flow cytometry. We used antibodies to identify the microglia in the flow cytometry readouts, and we also looked at antibodies against key markers of inflammation like tumor necrosis factor alpha, TNF alpha, iNOS, nitric oxide synthase, and ICAM1, another adhesion molecule. And what we found was that, yes, there were increased numbers of activated microglia after this wildfire smoke exposure, and that they were expressing more of these inflammatory factors. There was also invasion of, so those are the resident microglia in the brain, they were activated, but there was also invasion of neutrophils from the systemic circulation. So we saw PMNs, which is abbreviation for neutrophils, and they were in greater numbers, about 50 to 100% increased over control. And we also saw monocytes invading as well. One of the more interesting phenomena we saw, though, was with the endothelial cells, those vascular endothelial cells. We usually use this marker called CD31. It's a cell surface receptor that just it should just define whether it is an endothelial cell or not. But what we found after these exposures, and hopefully you're a little familiar with flow cytometry gating, but on the x-axis, it shows the level of expression of this CD31 marker. In the filtered air group, you could see there were some of these little dots represent different cells where it was elevated. Those are our endothelial cells. But after wildfire smoke, some of the cells were much higher in expression of this marker. We then decided what we would do is separate and analyze these groups separately, because it seemed like there might be a phenotypic change to a subpopulation the vascular endothelium. And that turned out to be the case. So the cells up here that were expressing a lot of CD31 seem to be anti-inflammatory. So after this 20-day exposure, they're trying to fix things. They're trying to shore up some defenses and eliminate any inflammatory response. But the population that was expressing normal levels of CD31, they were lower in population compared to the filtered air, but they were still very pro-inflammatory in the sense of expressing these factors like TNF-alpha, CCL2, and INOS. So turns out there's some recent publications that help us understand this, that the CD31 marker, also called PCAM, can be differentially expressed. And when it's higher, it tends to be healthier in a sense. So this is comparing people with Alzheimer's disease with people without Alzheimer's disease. And you can see that the levels of CD31 expression in the control healthy individuals is quite a bit higher than that of the Alzheimer's individuals. And it may also vary depending on where in the vascular bed, so arterial versus capillary versus venous. So very interesting finding, made us realize that it's probably not sufficient to just look at a single snapshot of exposure. You really need to look at the dynamic response over time, which really sets the stage for this next study. And here we decided we're not going to wait around for natural exposures. For one thing, it's kind of morbid to hope that California catches fire. Two, it's just logistically very challenging to have mice ready to go and have your team ready to go and have it not happen over a holiday. So we have a laboratory and we can generate, I can set things on fire, as it turns out. So we've got this little borosilicate tube, and the little ring around it is a ceramic furnace. And so we can set things on fire in this tube that then sends it into a exposure chamber, and we can monitor the concentration and size distribution. The sizes are all very much in the nano size range from about 50 to 300 nanometers, so very much respirable concentrations of dusts. And what we decided was, since the exposures we see are invariably intermittent, for several hours a day, every other day, that became our model. So we expose for 14 days, but it's day 2, day 4, day 6, day 8, etc. So every other day and 4 hours a day. And there's a little bit of fluctuation in the concentrations, but overall, our average for the 4-hour periods was under 500 micrograms per cubic meter. And our 24-hour average, because we're skipping days, is basically right around where we were before at about 37 micrograms per cubic meter. And because we're working with fresh emissions, we have also measured carbon monoxide as well as oxygen, oxides of nitrogen. And those are well below EPA standards for these two criteria air pollutants. Now, my graduate student, David Sheska, who did this, loves these Sankey plots. I love them too. They're really interesting in shape and help you describe the study. So we started with 30 mice in the wood smoke group and 30 mice in the filtered air group, and we exposed both for 2 weeks to either wood smoke or nothing. And then after that, we took a subset 1 day later and did a tissue harvest to look at the brains. 3 days later, we took another subset, 6 per group, 7 days, 14 days, and 28 days. So we have this snapshot where, or rather than a snapshot, we have 5 data points, I can count, of 1, 3, 7, 14, and 28 days of outcomes from these animals. All right, so I apologize. This is where things get a little complicated, but I think once you start to understand these graphs and you can look into the matrix, it becomes very clear. This is a radar plot, and basically the little dash line in the center is saying that anything above that line means there's a greater amount in wood smoke, and anything below that line means there's less in wood smoke as compared to the filtered air controls. So again, this is a CD31 high. These are those anti-inflammatory endothelial cells that we had previously seen. And sure enough, we're able to repeat that study and show that here we see greater percent in the wildfire smoke animals, and that they are showing a little bit less expression of this inflammatory mediator INOS. That's on day 1. on day 3 and on day 7 and on day 14, those CD31 high are still elevated. And to some extent, they still maintain an anti-inflammatory phenotype. But what's interesting is the medium expressing cells, which seem to be pro-inflammatory, they are still lower in concentration. There's fewer of those cells relative to filtered air. But you can see as you get to day 3 and day 7, the expression of inflammatory factors, especially the vascular cell adhesion molecule and CCL2, the chemokine, those are dramatically upregulated in this small subpopulation of endothelial cells. So what's neat here is that this is 7 days after we've stopped exposing them. And even out to day 14 and day 28, we're still starting to see effects. Now, when we also look at the cells, the immune cell population, we see similar trends. So this is activated microglia on the top, peripheral immune monocytes coming in, and then infiltrating PMNs. And just to kind of keep this short, I'll just point out that the infiltrating PMNs, you can see the circle kind of grows across the 7 and 14-day period. You also see that in the peripheral immune cells, where you see a greater response here at 7 and 14 days, and maybe even some residual dysfunction or dysregulation out on day 28. This is a whole mess of data, and I'm not going to go through it in great detail, other than to say that we also looked at metabolites using metabolomics approaches in the brains. And what we saw was that there was this whole 28-day period of evolution of the metabolic profile. And I'll just cut to the chase that shows this last figure sort of collectively sums up. We see this initial immune response from endothelial cells that peaks around day 7, that drives greater immune cell infiltration and activation that peaks 7 to 28 days or 14 to 28 days later. And the metabolic changes were acutely high, and they recovered, but then they started to trend upwards as you have this sort of latent neuroinflammatory response. Okay, so the last study I'll talk about is taking that same idea of the 14-day exposure period, but looking in older mice. So now, instead of healthy young 8-week-old mice that represent your average 18-year-old on the street, we're now using an 18-month-old mouse from the beginning of the study, and we're ultimately going to let them age to 21 months of age. And so this is much more reflecting of like my age, maybe out to 70 years of age in a mouse. So again, we have a period of 2 weeks where we expose them. We have one group where we euthanize immediately after the exposure. But these other groups, we let them age for 70 days more, so 10 weeks. And there's one subgroup where we give them control. There's another group where we give them a supplement of resveratrol and nicotinamide mononucleotide. The complexity of this study is quite high, and I don't have a lot of time, so I'm not going to dive into a lot of detail here. But resveratrol is a polyphenol in red wine that is supposed to make us live forever. Nicotinamide mononucleotide is a precursor to NAD, which is a very important energy molecule that your brain and your heart need. So those are the two main groups I'll talk about. We also looked at dasatinib and quercetin, which is a senolytic cocktail to try to stave off senescence in the brain cells. And then the last is a combination of all of these things. But don't worry about those so much. We'll focus on the nicotinamide mononucleotide. And really, so this is a paper that we have submitted right now. It's actually in preprint form to get into all the nitty-gritty details of the metabolomics. But the key finding that is worth sharing is that in the prefrontal cortex of these animals, we measure a lot of different metabolites, and serotonin, which is highly associated with mood, was not quite significantly reduced 1 day after the exposures, but 10 weeks later, it was absolutely significantly reduced. We also did a behavioral study in this. It's a forced swim test, and what we found was time immobile was reduced, vice versa. basically there was a greater expression of a depressive phenotype in the mice exposed to wildfire smoke that lines up with the serotonin effect. And it was sustained throughout the exposure or beyond the exposure 10 weeks. Optimistically, the resveratrol and nicotinamide largely fixed that, as did the senolytics. So combined, there is an effect of wildfire smoke in older animals that may be a long-term concern, but it may be that there are supplements and or dietary factors that you can boost following the exposures that can help to normalize that. All right, I need to get a better figure for this. This is all fuzzy. We also looked at cardiac function in these animals. And if you just look at the three clusters on the first 6 bars, if you will, you're looking at young animals, 3-month-old animals, filtered air and wildfire smoke next to each other, and then the 18-month-old animals, and this is again 1 day after the exposure, but the same exposures, and then 70-day post, this is again filtered air and wildfire smoke. These 3 combined have no other treatments, so when you put all that data together, there is a small, subtle effect of wildfire smoke in reducing ejection fraction, and it's also consistent with stroke volume. The last 6 bars are basically showing what happens with the supplements, resveratrol, nicotinamide mononucleotide, and the senolytics. For the most part, those normalize that, although the RNDQ, the combination of everything, seem to be far more detrimental, actually. When we looked at the metabolomics in the heart, it was very interesting that 3-month-old mice immediately after the exposure had a response of metabolites. And so these are volcanoplots. Basically, anything above the little dashed line shows the significant metabolites that are changed. And you can see there's a couple dozen of those. In 18-month-old mice, there were far more, probably 3 times as many in the 18-month-old mice. And when you waited 10 weeks later without any treatment, there's still quite a lot of sustained metabolic changes in the hearts of exposed mice compared to filtered air. And when we look at the resveratrol and nicotinamide mononucleotide and senolytics compared to that sustained effect in vehicle-treated mice, you can see that practically no significant changes are left in the resveratrol and nicotinamide mononucleotide group. Some in the senolytic and the other group are still remaining. But this, again, points to the fact that wildfire smoke may have some detrimental effects on the heart as well as the brain, but that there might be opportunities to offset that with supplements and diet. And I'm gonna, in the interest of time, cruise to sort of this last thought, which is a very new paper coming out of Michigan, University of Michigan, and some collaborators from Washington and elsewhere. But this is Sarah Adar, is a fantastic epidemiologist, and she published this in the last couple months, looking at exposure to particulates and incidence of dementia. And when she did the modeling and controlling for all the factors and the multi-pollutant exposures, what came out of this study was that the strongest, most consistent effect on incidence of dementia was exposure to particles that arose from wildfires. So again, it sort of points to the fact that there may be some unique aspect of wildfire smoke that is detrimental to the brain, and it could have acute and long-term outcomes. And I think my big take home at this point is we really need more studies like Sarah just released, because we absolutely are limited in what we know about sort of the distribution of wildfire smoke and its impacts and the intermittent nature. I think if you live in a modern urban city, there's a background of PM from traffic and other sources that your body has a chance to get used to. But when this wildfire smoke comes across and causes much higher levels for days to weeks and then goes away, your body doesn't really have a chance to recover from that. You saw how long it takes for the brain to sort of reset itself, even in healthy animals. So just in summary, this is a problem that we're facing where we've increased the wildfire smoke that's generated, the acreage burned over the past 40 years, probably due to changes in our climate. Obviously, respiratory and cardiovascular implications are of concern, but our preclinical studies and certainly a couple of really nice epidemiological studies raise significant concern about neurological impacts, both in terms of learning, memory, mood, as well as long-term risk of neurodegenerative diseases. Our work has really been focused on understanding the cellular and molecular basis of that neuroinflammation. But there's still a tremendous amount of work that needs to be done understanding the real behavioral implications of that, and even further, what can be done about it in terms of, diet, supplements, things that can help people avoid these outcomes. And that brings me to the end. So I just like to thank the wonderful people who really do the work here. This gentleman up here is David Sheska. He just graduated, so that's Dr. Sheska. He is going off to a job at a startup company in California. Jessica Begay here does a lot of our wildfire smoke exposures now. She's been on the team since she was a master's student, and she is just an absolute warrior for this work. This gentleman down here, Andy Ottens, has been key to understanding the peptide story, understanding that this molecular shrapnel extends from the lung to systemic organs like the brain. So with that, I will end the talk and just say thank you to everyone, and happy to answer any questions.
Yeah, thank you, Matt. We appreciate the opportunity. Thank you for this interesting talk. If you have any questions, you can post them on the chat, and then I will ask them. I have a question. So all these responses, are these due to the species or the pieces of protein, the sharpnel that you identify, or have you been able to see if any smaller particles translocate into the bloodstream and then they cross into the blood, or you think that these responses are predominantly due to the these components of proteins?
So I think I have limited concern about this sort of direct translocation model, and there's a number of reasons for that, but probably my biggest is that I can replicate all of these findings with ozone. And ozone is such a reactive molecule that it won't pass more than 1/10 of a micron into the liquid layer of the lung. So ozone causes secondary responses that propagates, metalloproteinase activation, peptide formation, and then neuroinflammation. What we haven't done, partly because I know that I'll get shut down by so many people, what we need to do is direct IV injections of particulates to show that, yes, they get to the brain and they do or don't cause anything. But I at the end of the day, I think people can wrestle over that all they want. Doesn't change the fact that there is a neuroinflammation, and we should probably figure out something to, dial that response back somehow.
Okay. So there is a question, since you mentioned that with foods and dietary supplements, we can address a little bit of that. So what foods and dietary supplements contain the components that help buffer against the damaging impacts of wildfire smoke?
Yeah, it's a great question, and we've been limited in what we can look at with our ozone studies, where we're really focused more on fetal maternal outcomes. We've been using polyunsaturated fats and comparing that with high saturated fat, like coconut oil versus soybean oil. And what we find is that the saturated fat tends to make things really worse in a variety of ways, whereas polyunsaturated fats dampen the overall inflammatory response and create a better outcome. I think there's a couple of reasons for that. One is the sort of initial reactions in the lung are different because of those different fatty acids. but also systemically, you're poised to handle inflammation differently. Resolvins are small lipid mediators that help tamp down inflammation and hasten the accelerate the resolution of inflammation. And those can be created from the polyunsaturated fats. So what do I mean by all this? Well, we're talking fruits, nuts, avocados, the healthy fats, right? Trying to avoid the heavy meats and cheeses that contain the saturated fats. And my hat's off to our firefighters who are dealing with these wildfires, because I personally, after 12 hours of putting out fires, I would probably love a green chili bacon cheeseburger, but a salad, kale, good healthy fruits and vegetables is probably going to be better for you in the long term.
Okay. So I think, you mentioned that during the study, all these results have been published and they're available through PubMed and so on. So there is a couple of more specific questions. Is there a region-specific effect for the microglia activation following the exposure in mouse model?
It's a great question. So what we've seen is that the microglia very, very close to the neurovascular unit. So as that blood-brain barrier breaks down a little bit, and it's not like massive hemorrhage. It's just a couple of proteins getting across from the blood into the brain parenchyma. That triggers reactions from the astrocytes and the microglia. And it is a very focal response. Andy Otten's had a great paper where he sort of zoomed in there and he looked at a penumbra about 50 microns away from the neurovascular unit, and he saw a very clear dose-dependent response in activation of those of the microglia and the astrocytes. What we have not done, which the question may be getting to, is have we looked at, say, the prefrontal cortex versus hippocampus versus cerebellum, and we really haven't done that in a head-to-head manner. What I will say is all of those regions in one study or another have been affected in a negative way. And so we don't think that there's tremendous difference from one region of the brain to the other, but it does seem to be focused in and around the neurovascular unit.
Thank you. I think you already answered the next question. Does the time of exposure and the dose impact the cell types affected in the brain? And is there an activation of cell types which is region specific, or as you mentioned?
So dose is an interesting thing. I don't know that there's a huge dose response. I would assume that there would be some. In our multi-well carbon nanotube studies, we had a clear dose response. But what was interesting is I've seen data recently from Luke Montrose, who's up at Colorado State, and he's asking the question not for the general public, but for wildfire fighters. And the firefighters are looking at magnitudes greater exposure. So he's, instead of doing 0.5 milligram per cubic meter, he's looking at 40 milligrams per cubic meter of exposures, far higher than what we do. But he sees a tremendous commonality of effects. It would be nice for, if we were to do this and do a like 3 or 4 dose group, that would be really worthwhile. But I honestly think that what happens is you have sort of a trigger that gets turned on because of the blood-brain barrier deficit that then, causes a cascade of effects. And there may be some gradations of the level of response, but to a certain threshold, maybe it stops at a third. It's a good question. I wish I knew the answer. I guess is the upshot.
So you mentioned the metabolomics profile changed over time. Are there any specific metabolites that
Yeah. So the first thing we've been looking at is NAD, which is a major energetic metabolite that does decrease as we age. That's probably one of the biggest factors associated with mental loss, cognitive loss in old age. So we've been looking at that, and absolutely in every study we've done, regardless of sex or age of the animal, wildfire smoke reduces NAD acutely. In the young animals, it does seem to recover 28 days later to a level higher than it was before. We don't really see that with the older animals. It does recover though. We've also looked at kynurenine, which I don't know much about, but it's definitely altered across that time course. Then there's a couple other metabolites. I've been approaching it from the gestalt, the whole
changes, yeah.
picture. But we're starting to dive into the data now and try to understand more specific details of those pathways.
Okay, thank you.
Mhm.
I think that completes all the questions. I appreciate all of you asking the questions and get more information and more details. With that, again, I would like to thank our speaker for joining us all the way from Albuquerque, New Mexico. It's the place for chili, if you don't know. So, and thank you, Matt. Thank you for joining us, and thank you for coming in this grant routes for the School of Public Health. I think the presentation will be available on our social media later today or tomorrow.
Well, thank you very much for inviting me. I've had a great time and appreciate being able to share our research with you.
Yeah, it's quite famous the last few months. Thank you, Matt. Appreciate it.
Take care.
Thank you, everybody.